Okay.
Hello, Ralph. Ralph, you're muted, but it's okay.
Now it should work.
Yes.
Since I use so many different of these systems, I never know what I need to do to share my screen.
So we have a couple of minutes. Would you like to try that?
Yeah, maybe. So I have a button here. If I click that button, what will happen?
So just for the benefit of the other Ralph, no, the first speaker that just joined us. So Ralph Everest is going to try to flash
we are recording now.
Systems. I never know what I need to do to share my screen. Yes. No.
So we have a couple of minutes.
We are recording now. If you don't mind, we can try at the break.
Yeah, click that button. I have too many windows open. Now you have to tell me if this works.
Okay, yes, we see your desktop. So
you see the presentation screen as it should be. Okay, very good.
We see the presenter screen.
Mm-hmm.
We have 1 minute, Ralph. If you want, then we should switch to Ralph Eichhorn.
I'm second, right? Yeah. Is this the way it should be or not?
I don't think so.
Yeah. You're the second Ralph, yes.
Okay. Then I should just stop doing anything, right? So,
okay.
Where's this Zoom thing? Stop share.
Okay. Thanks, Ralph. I think we have a break, a quarter of an hour break after Professor Eichhorn talk, and we can figure things out there.
Too many Ralph E in one session.
Right. Okay. So now I will mute everybody and unmute Professor Eichhorn. Okay. So Ralph, are you set to go with the presentation?
Yes, right. I'm ready.
So welcome everybody to this afternoon session of the SECAM School. My name is Cristian Micheletti, based in Sissa, Trieste, and we have 2 talks in the afternoon session. The first talk will be given by Professor Ralph Eichhorn from Nordita in Stockholm. The second talk by Professor Ralph Everest, based in the École Normale Supérieure in Lyon. So the first talk, Professor Eichhorn, we start momentarily, and he will talk about Brownian dynamic simulations with hard-body interactions, exact numerical treatment. So Ralph, to you.
Yeah, thank you very much. I also would like to thank you, the organizers, for the invitation. I'm happy to join this meeting. Of course, I would have been more happy to be actually in Trieste right now, but that's how it is. So I start sharing my screen now. Okay, you can see it, the presentation, right?
Yes.
Okay, good. Yes. So as said, the title is Brownian dynamic simulations with hard-body interaction, exact numerical treatment. This is a talk basically about these two papers I published together with Hans Behringer, who was in Mainz at that time, but he is now in Hamburg. And this is, in principle, it's a surprisingly simple problem which caused us a lot of headache. And if you go through the literature, there are many attempts to solve it, but they're not really correct, all these attempts. I will explain a little bit more. So these are about Brownian dynamic simulations, Brownian motion. It's a particle in suspension or interacting particles in suspension in an aqueous suspension. There are external forces that can be driven by electric fields, by fluid flows or whatever. And important here, they are moving in a structured environment. So that there can be obstacles or confinements, chambers. There are no limitations to your fantasy. And here, what I'm talking about, this kind of simulations, it's not directly a molecular dynamic simulation. So I'm not treating the solvent explicitly. The solvent here is modeled by viscous friction and thermal fluctuations. So these are simulations on the level of so-called Langevin equations. I will show the equation in a second. These kind of simulations with a structured environment and external forces, they have many applications. In particular in microfluidics, where, for instance, I did a lot of simulations in trying to model many experiments, and this is one of the structures I simulated a while back. Then you can apply to biomolecules in the cell, self-assembly polymers. Always when you model these objects more or less as heartbeats, or maybe objects that have a little bit more structured shape than just spherical. I will focus on spherical objects here in my talk, and it's about the numerical simulation of these systems. So how do you do the simulation if there are these interactions with the hard walls? For instance, these obstacles here. This is a particle interacting with these obstacles driven in this field. And the model is, as I said, a Langevin equation, which I've written down here. R dot, first time derivative, is equal to the forces on the particle. This is just for one particle for now. And here's the diffusion term, thermal fluctuations, which models the thermal noise coming from the environment. And this is the friction coefficient. And the fact that this here is a first order time derivative in this force balance tells you that we already have neglected inertia effects, second order time derivatives. So we are in the overdamped limit. This is a very common idealization which is used in this modeling approach, and it's very, very well fulfilled. So there's an order of magnitude difference between inertia effects and friction effects here on this micrometer or nanometer scales in aqueous solution. So this is one of the common idealizations we use. And the second one is that interactions between bodies, the extremely short range and strong repulsive contact forces, are usually modeled as hard body interactions. So there are no forces when the objects don't see each other, and when they touch, the force is more or less infinite at the contact point. And for that reason, these forces are singular and cannot be included in this force term here. So these are external forces or maybe other kind of interaction forces between the particles, but not these contact forces, because they are not regular enough to be treated in this kind of differential equation. And this causes a problem when you want to do a simulation. Standard is to use the Euler algorithm. It's the simplest discretization you can think of, and we will stick to it for the talk here. So you calculate the displacement at a certain time, from the forces acting at this time. So here I abbreviate it as a velocity, because force divided by friction coefficient is like a velocity, it's a deterministic velocity coming from the external forces. And here is the term, the discretized term from the thermal fluctuations. And this GT thing here is a Gaussian, or is a d-dimensional vector, this dimension of Gaussian normal distributed variables. They model the thermal fluctuations. And then you simply update. The new position is the old one plus this displacement. That's all fine and works very well as far as you don't see any walls. This here is a wall. You don't see it yet. But what are you going to do if you do such a step and you end up in an end position which is not a valid configuration? You end up in an obstacle, in a wall, or overlapping with other particles. And if you want to treat this in a numerical method, you need a kind of algorithm which tells you what to do. And the first part of this algorithm is that you have to detect these unphysical configurations, these collisions, but keep in mind, they are not really collisions like elastic collisions, because this is an overdamped diffusive motion. So a particle is diffusing here and then touching the hard wall at some point and diffusing away. It's not an elastic collision that happens here. Anyway, I call it collision because it's such a common term. I put it in quotation marks. So this is one part of the algorithm. You have to detect these unphysical configurations. And this is a geometrical problem, mainly depends on your structure. I'm not going to talk about that. I'm going to talk about the second part, about the rule you have to apply to generate a physically valid configuration from that situation. So what are you going to do if you encounter such a situation to make it into something that is consistent with the equation of motion and the force fields acting and this hard boundary here? And of course, this is a very common problem when you do simulations. So there are many articles on that, trying out different things. And all these methods have in common that they somehow are heuristic. They come up with some rule and then apply it and try if it works or not. And there are two very prominent examples which you can also use when there are external forces. When the deterministic force is 0, then things are a little bit simpler and there are even more heuristic methods, but I'm going to focus on those where you have a deterministic external force. And the simplest one you can think of is a rejection scheme. That is, you simply discard this unphysical configuration and try out a new one by generating a new Gaussian random number from this thermal noise and try out if it leads to a configuration which is actually okay. And then there's the question, what are you going to do with the time during that step? Do you advance it or not? So here you already see there might be a problem. It's just something a simple thing you can think of. There's one reference here where they suggested it, but there are many more. And then another one is a more sophisticated method. It's an event-driven scheme. Here the idea is that you propagate the particle only a fraction of the time step until the collision point where it actually hits the hard wall. And then you use the rejection scheme for remaining time step. So you somehow simulate one collision with a wall, but if you know a little bit Brownian motion, it's quite clear that this also cannot be fully correct, because in Brownian motion with a white noise, a particle can cross a certain line infinitely often in any time interval. So this here is just one collision. It's also like just an idea. Okay, let's try out if it works. It works actually surprisingly well. I will show a little bit later. But all these methods, they lack actually a thorough justification. They are rules of thumb, you can say. You try out and you see if it works or not. If you what if you really want to do it, say, correctly? What are you going to do? Is there a way to do it correctly, let's say, or with a justification? And this is something which took us, Hans and me, took us a while to realize. In fact, in the end, it's very simple. But if you think, what does the Euler algorithm actually do? It creates a new position here by using this Gaussian random number and advancing from the old to the new position that corresponds to a probability for this displacement, which is a Gaussian, simply because this GT here is a Gaussian. And this Gaussian is actually a diffusive displacement by dr, and the center of the motion drifts along with this velocity value, which is constant constant during this time step. This is the discretization. Even though the force depends on space, for that time step it's taken constant. So the Euler algorithm actually generates a new position from the transition probability, the probability to be at position r t plus dt when I have started out from a position r of time t. So it's a step in the transition probability. The Euler algorithm chooses a new position from the transition probability. Okay, and once you have realized that, you simply can say, okay, all we have to do is we have to generate a new position when there is this wall from the transition probability, which actually takes into account the presence of the wall. Okay, that sounds simple, but in general, we do not know. If we knew that, we wouldn't have to do the simulation, right? So we do the simulation because we cannot solve the equations of motion. We cannot write down the transition probability. So it seems that this is it's almost hopeless, and this is the reason people come up with rejection schemes and so on. But since you're discretizing anyway, like the force is constant, if your discretization is small enough, you always have the situation that you're seeing locally a flat wall, like you're seeing locally a constant force. And close to a flat wall, you can, yeah, this is the question, what is the transition probability? And close to a flat wall, you can decompose the motion into a perpendicular and a parallel component, which are uncorrelated. Statistically, you can show that. And then the collision only happens in the perpendicular component. The parallel component is completely unaffected. Imagine the displacement would just suggest the parallel component, then you would not even see the wall. So the collision happens in the perpendicular component. And this is the one-dimensional problem. So we only need to know the transition probability for a one-dimensional motion where there is a reflecting boundary. And this is actually known. This has been calculated by Smoluchowski already in 1916. It's the driven diffusion on a half-line with a constant force, which is here in the velocity. This is the diffusion equation. dq is the diffusion coefficient. I call this coordinate now q, this one-dimensional coordinate. And there's a deterministic velocity vq, and the wall is represented by a reflecting boundary at the origin. So there is the current, the probability current through that origin is 0. And the initial position is some q naught at time t equals to 0. And then Smoluchowski has calculated that the full solution looks like that. It contains of 3 parts. And here I've written down the 3 parts. The details are not so important for the moment. Important here is that P1, the first part, is actually exactly the free diffusion part, as if the reflecting boundary is not there. And then you have 2 parts which are somehow corrections to that. But keep in mind that this solution only is valid for positive Q. Okay, so in total it's normalized for Q positive, while the P1, if it's free diffusion, is also normalized over the whole real line, but the 3 together are normalized over the positive Q. So we know the transition probability, exact transition probability for any time when there is a reflecting boundary at the origin. And this we can use. And I will show you a few pictures here of this solution. The solution is always the black line. Here are the parameters. So VQ negative means that the deterministic force is pointing towards the origin, towards the reflecting wall. And the dashed and dotted lines are an attempt to simulate this motion in 1D close to a reflecting wall in the origin using the rejection scheme. Okay, so any time we find the position hitting the origin or being beyond the origin, we just discard the step and try a new one. And the distribution that comes out for dt equals to the whole time I elapse the motion here, t equals 0.05, is the red dotted line. And then if I decrease dt, the integration time step of the rejection scheme, by a factor 10, it's the blue line by a further factor 10. So in total by a factor 100 is this dashed dotted line. So you can see that it never really gets the distribution right. And here is the same situation when you advance time while rejecting the steps. And there you see there's a kind of delta peak at the initial position. And this is the one where you have that you have rejected, and you just stay there, and time is further advancing. And this insert here shows an enlargement here of the event-driven scheme. So the event-driven scheme for dt equals to 0.05 is very, very good. It is on the large scale, it's indistinguishable from the black curve, but if you zoom in, you can see a small deviation. And I should add that for these parameters, the collision probability to hit the wall, to hit the origin, is actually only 8%. And still the rejection scheme is quite bad. So you see, you may have a problem if you use this all the time. There's another example here, which I skip because time is running. So I come back to this solution. As said, I have the free diffusion here and some correction. You see also that here, for instance, there's a negative sign. That's not a typo, that's correct. So for positive VQ, forces pointing away from the wall, this contribution here is actually negative. So you can also not correct by simply adding stuff to a first attempt, because there's a negative contribution here. It's more complicated. So the idea is, okay, because we have the free diffusion part here and then some correction, the idea is perform standard integration as long as the displacements do not lead to unphysical configurations. Okay, the particle doesn't see the wall, just go on and do your standard integration. If you hit the wall, if there is a collision, replace the component along the collision axis, so which is involved in collision, this one-dimensional direction Q, by a new displacement, which is drawn from this distribution here, the P2 plus P3 normalized. And the W here is the integral from 0 to infinity of P2 and PQ3, rewritten in that way just by using normalization. And this is like the collision probability, if you like. So in case you encounter collision, correct your dq displacement by using a random number drawn from this distribution. Okay, so why does that work? There's a tiny little calculation here. So the collision axis, the one-dimensional direction where the collision happens towards the wall, this is this half line from the Smoluchowski solution, this coordinate q, with a collision point at the origin. And the random number created by my algorithm on this collision axis is a q, which is Gaussian if it's positive. This is the Heaviside theta function. So if this q Gaussian is positive, I also take it, I keep it, okay? I said, then I didn't hit the wall, so I don't change anything. If it's negative, meaning I went through the origin, my end position is inside the wall, I take a q which is taken from this collision probability, from the PC, which is P2 plus P3 divided by omega. And then I can calculate the distribution, which I actually get from this approach by my algorithm, just transforming usual transformation of probabilities, variables. There's the Gaussian for the Q1, and there's the collision probability for the Q2. And then this delta function enforces these constraints that I keep Q1 when it is positive, and I take it to Q2 when Q1 is positive, negative. So I can do the calculation, and in the end it comes out, yes, this is exactly P1 of Q plus P2 of Q plus P3 of Q. So this algorithm correctly reproduces the Smoluchowski solution. That means that if the force is constant, the Smoluchowski solution is for a constant force, I can do an exact integration step close to wall with an arbitrarily large time step, because I have an exact solution. I have an exact transition probability. And this is what I'm going to exploit here. As said, the collision direction is this dr perpendicular. So I go on with my integration using standard Euler scheme. If there's no collision, maybe I have to do some correction. This is the reason I write here now an drt star. This is my final step I really accept after the algorithm has been applied. So if there's no collision, I just can use the free Euler scheme. If there is a collision, I have to do a correction with this q minus q naught, with this dq here taken from the Smoluchowski P2 plus P3 distribution. And then this n here is the normal vector at the wall. So this actually here I subtract from this displacement I have calculated before, I subtract the normal component and I add the new dq. This is basically what happens. So I take out the normal component because it's not correct, and I replace it by a correct one. And this is done in that equation. So this is not such a complicated equation, and the algorithm in the end is quite simple. Important here that I can do it is that the dr perpendicular and dr parallel are uncorrelated, because I changed the statistics of the perpendicular component. If it were correlated to the parallel component, I would have to do something here too to keep the correlations. But one can show that these components are uncorrelated, so everything is fine. So that's also the idea, if I want to generalize, that I look for coordinates which are uncorrelated statistically. In the same way, one can treat collisions between two particles. The end formula are these here. This is interesting because imagine I have a collision with two particles which are not of the same size, so which feel different friction. It's not really clear how to, from the very beginning, it's not really clear how to separate them so that that everything is correct statistically? Do I have the larger particle a little bit, move a little bit less and the smaller a little bit more, or what do I have to do? And this formula tells you how. It's related to the ratio of the friction coefficients. And this E vector is actually the collision direction. So this formula can be derived by transforming the whole motion to the center of mass coordinate and to a relative coordinate. And then you show that these coordinates are not correlated. And then in the relative motion, we correct the direction which is connected to the collision axis of the two particles. So the idea is the same. And the final results after some calculation are these formulas. So yeah, I don't want to say more. You can look it up in the papers, and I would try to give you the idea how to do it. And this is, there are many other examples, but this here I like because you might think, okay, you have the rejection scheme or the event-driven scheme. I just make the time step small enough, and then the distribution may not be fully correct, but I don't care. I don't need the full correct distribution very close to the wall. The thing is, sometimes it does not work at all. For instance, and this is how I stumbled actually into this problem. Imagine you have a particle which is driven by the deterministic force a little bit into the wall, and you're interested in the escape of the particle from a certain region. So it moves that way and diffuses at the same time, and at some point it will cross that gray line. And this is my goal. This is where I want to go, and I want to measure the time it takes. I want to measure the mean first passage time for the particle it takes from here until it crosses that line.
Ralph, let me tell you that you have 5 minutes left.
Okay, thank you. If you do that simulation here, then the mean first passage time is that line here as a function of the velocity, deterministic velocity, so the driving force in the end. Here's the driving force. It's pointing into the wall and the component parallel to the wall. And the velocity is F divided by friction coefficient. And this is calculated for a particle radius of 1 micrometer in aqueous solution at room temperature. And this is the integration time step. And here you see the mean first passage time. Calculated by our algorithm and also by the event-driven scheme. You cannot see a difference here. So the event-driven scheme is fine for this problem, but the rejection scheme, which is the blue dotted line, gets it completely wrong for large forces. And the reason is that the particle basically gets stuck in the wall. This force is so large that almost all the attempts you make to find a new displacement end up in the wall, and somehow you get stuck. So your whole simulation stops. And you get times also, if you're thinking you want to simulate association times of molecules, you get them completely wrong. Another thing here is instead is the computational time in arbitrary units. The black line is our algorithm, which makes use of the exact solution, so it doesn't waste time in a way. And this dotted line again is the rejection scheme, which also goes up because it has to throw away a lot of steps. And the dashed line is the event-driven scheme, which gives the correct value for the mean first passage time, but the computational time goes up very much for large forces because it also has to discard a lot of steps in the end and has to repeat a lot of steps. It's two curves because there are two slightly different variants of the event-driven scheme, which I don't want to go into detail now, but you see that from a computational efficiency perspective, that here the correct method of course gives you an advantage. Okay, in practice, a few words. You need detection of collisions, as I already said. You need that for any integration scheme. I have not talked about that, and this is a geometrical problem. That may be quite difficult, actually, but this is something you have to solve independently. The integration time step in our method is determined by variations of f and by curvature of structures or particles now. So you discretize your integration. You're usually used to choosing a time step small enough so that over a typical displacement, the deterministic forces are virtually constant. Then your integration scheme is fine. And now, since you're also simulating structures, the structures have to appear flat on that scale, straight, flat walls. So that's a second constraint you have on your integration time step dt. But it's like the usual discretization you do. You just have to choose it small enough for the system at hand. Another thing is how to generate a random number according to this weird distribution, which involves error functions and so on. And there's this method, I think we found in numerical recipes. You calculate the cumulative, and the inverse then of the cumulative gives you, with a uniformly distributed random number, gives you a random number which is distributed according to your distribution you want. The thing is that this function you cannot invert analytically, so you have to do it numerically. For instance, you can use some scheme from the GNU Scientific Library, and this slows down the simulation a little bit, but you still have seen, as I shown before, that it's still faster than the other methods. Okay, and that's sort of my summary. That's a part, what I presented has been some part of these two papers. And we have been ever since, you see, it's already almost 10 years ago that we did this. We have been ever since thinking about extension or generalizations, but it turned out to be very difficult. For instance, what happens for non-spherical particles? There you have rotational degrees of freedom 2, and once they collide, you have to distribute this correction you do, this re-evaluation of the collision axis, you have to distribute the new position over the rotation and the translation in a correct way. And that's not so easy to figure out. So we don't know yet. And then what happens in corners, or if you have like wedges in your structure, because there you could also say you have like an infinite curvature. So then the algorithm in that way also does not work. What happens if you have really many particles, so that there are not only like 2 particle collisions, but 3 or 4 particle collisions in one time step dt? Of course, you could decrease your time step dt and make it smaller and smaller and smaller. So this happens rarely, but then you lose the advantage of that algorithm, because then you have to, again, you have to go to very small time steps. So it would be good to have an extension of the algorithm which takes into account, say, 3-body collisions or even 4-body collisions. But honestly, I don't think that this is possible, at least not on the fully correct level we did it here. Okay, thank you very much for your attention.
Thank you, Ralph. So you can join me thanking the speaker. Thank you very much for being perfectly on time, and especially for this very clear and didactical talk. I think it's perfectly suited to the school. So we can have questions in the chat. I will read them out for you. Just to get started, let me ask a question. It's the first time that I hear about this topic. It's really fascinating. So I was wondering regarding the more challenging problem of having extended anisotropic objects, having to deal also with rotations and so on. I understand the challenge. How about existing algorithm? Let's say, is there an event-driven scheme that does this job of distributing the rotation and so on, or no, it would be a first algorithm ever to do that, the one that you're working on?
No, it would not be the first algorithm ever. Actually, you use the event-driven scheme that I discussed here, because let me go back. This event-driven scheme just idea you create until the point where it collides. And this you can do no matter what how your particle look like and how they rotate and how they move. And then you have to think about what you want to do then with the remaining time step. And the question is also, of course, does that lead to the correct distribution? I don't know, or at least to a distribution which is close to the correct one, let's say. I don't know, but I suspect yes, because what I did not discuss, what we can show actually, that this event-driven scheme correctly captures the P2 contribution of the Smoluchowski solution. So the P2 contribution can be associated to just one collision during the time interval t, and this is correctly captured by the event-driven scheme. So I suspect that this would be a good start to do, but it's also numerically expensive, because to find that collision point, you have to do like a root search, like more or less exactly what I have to do for finding the random number q. You have to do numerically to find that collision point, because you cannot solve it analytically in general.
Thank you, Ralph. So we have some questions starting to appear in the chat box. So the first one is from Samandata. Thanks you for the talk, and then asks, is it possible to extend the analysis for fluids in channels or other types of confinement? If yes, what modifications one needs to follow?
There are basically no modifications. You can do it directly if in a simple situation where you do not take into account hydrodynamic interactions between particles or between particles and the wall. So if your system is such that you can model the fluid flow in the channel by just, say, a velocity field which drives the particle, then it's exactly this here. And then you have the thermal noise as usual. And the walls, the channel walls, you can represent by some geometry. But this is then what I said a few times. Then the task is also to detect the unphysical configuration. So you have to have a part of your numerical, of your simulation algorithm has to tell you what are the positions outside my channel. This is what you have to know. And for applying the algorithm I suggest, you also have to know the distance from the wall and the normal at the collision point. This is what you have to know to use that algorithm. But you can apply it directly, exactly like I described it under these, if you don't want to include hydrodynamic interactions.
Okay. So while we wait for further questions, let me ask, is there a publicly available code or implementation of your algorithm?
Oh,
that
no.
you can try out and play with?
Unfortunately not, but anybody who's interested, please drop me an email and I can send you the code. I have it in C, and it's a very small, it's actually, when you use libraries which can handle vectors and so on, it's a very simple code in the end. It's very simple. It's not.
Okay, so it's a plugin quite transferable from code to code. Very well. So other questions from the students?
Can I ask a question?
Yeah, please go ahead.
Well, thanks for the great talk. I have a question which is the following. So can you extend the algorithm in the case of space-dependent diffusion, even in a simple example?
Yes, I think so. Then your discretization is also to be like small enough so that the diffusion coefficient doesn't change significantly over the typical displacement. But otherwise, it's fine. And then, of course, you have to take care of of the multiplicative noise. I mean, you have to know which kind of interpretation you're using. If it's E2 or Stotanovich in the Overdamm case.
Yeah, another unrelated question. So you can reproduce the mean first passage time, but is the algorithm able to reproduce the full distribution of the first passage time?
The mean first passage time, what was
it? Yeah.
Yes. Let me see. I think we have a picture in the publication. Let me see if I can find it quickly. No, it's this one. I think we looked at the distribution, at the full distribution. But the answer is yes, it can. We have lots of examples here, very interesting. No, it's not in here, but we tried that. So the algorithm can reproduce the first passage time distribution, yes.
Okay, thank you.
Thanks. So other questions? There is a further question from Samandata, and meanwhile I invite others to ask other questions if there are. So my second question is, if we add some activity which may come from any means like shape asymmetry or orientational fluctuations, etc., would that be working in the same way? Because adding activity would be just like adding one more noise term. And okay, and Samandata would be interested in receiving the code. So please contact the speaker for that. So Ralph?
Yes, if you can represent activity by an force term, by an external force term, which enters here, like for an active bounding particle, you would have rotational diffusion and force which depends on this directional diffusion. You can add it here, then it would work exactly the same way. Yes.
Okay, yeah. Okay, questions from others? So I think the chat box is complete. So thank you, Ralph. Thank you for this very clear talk. Join me in thanking the speaker again for the
my
talk.
pleasure. Thank you.
And now, of course, participants may contact you for follow-ups about the code or other details. I hope you are available. And we take a 15 minutes break before the talk of Professor Ralph Everest, who has already joined us. Thank you. Thank you very much again. So now I'm asking the ICTP technical support if we should try out the setup of Professor Everest.
Okay. So first of all, we would like to listen to the microphone, but I think it was working before. So, and also before you start that lesson, this lesson that ended now, I've seen that he already shared his presentation, but it missed.
Yeah, but somehow it was in presenter mode. We didn't see the full screen one.
Well, let me just try again, maybe.
Yeah. No.
Looks okay. Looks okay.
Yeah,
Then, all right.
Yeah.
Okay. Because now I'm sharing a second screen, and this is like this is where this PowerPoint thing goes, and I didn't know how to do that first. So I think.
So Ralph, I will let you when you have 5 minutes left to the end of your talk.
Yeah. The other Ralph, are you still there?
Who?
Ralph Eichhorn.
He was a minute ago.
Yeah.
Yes, he's still there. Yes,
Still there.
still here.
But he doesn't hear me.
I'm here. So
Ah,
now,
you're there.
yeah, now you can hear me. Yeah, I'm
Yes.
still here. Yes.
Very nice.
My video.
Stupid question. I mean, this whole collision problem is infinitely simpler for ballistic motion, right? Because the particle doesn't cross the boundary several times, right? So in that case, I think you can just reflect its position with respect to the boundary and then, or do a little bit more if there's an external force. And so that's much easier to handle.
Exactly.
And so I've just been playing for my lecture with multi-particle collision dynamics.
Mm-hmm.
And in that case, the streaming step would be sort of ballistic, right? So then I handle the boundary conditions after the ballistic streaming step, which is not so difficult. And then sort of this diffusive behavior would emerge from the collision step.
Right.
But the collisions would always, by definition, take place, I mean, in the good part of the system, because, I mean, I've put the particles back where they belong. Okay, so I've 4 weeks experience with this when I started programming it, but it intrigued me as an algorithm which is simple and maybe yeah, you can avoid all sorts of problems.
Yes, but this algorithm simulates the solvent, right? These are the solvent particles. It's a way to represent the solvent.
Yeah,
and then you have to embed something in the solvent. So when I want to simulate the Brownian motion of one particle, I solve one Langevin equation. If you want to simulate the Brownian motion of one particle, you solve, I don't know how many equations.
Yeah, and that sounds interesting.
I agree. I mean, all these methods, of course, if you need to have hydrodynamic interactions and things like that, then they are much better than a Langevin approach, I think. So then, because it's a mesoscopic way of simulating the solvent. You can do DPD, you can do multi-particle collisions, you can do lattice-Boltzmann. I think these are the 3 most common ones where you include the solvent in your simulations on a mesoscopic scale. Yes, it depends on the question. It depends on what you want to know about your system. Yeah.
As to for a single particle, you would.
Yeah.
Well, it's a coffee break, right? So. Daniel, ich bin gleich dabei, den Vortrag zu halten. Tut mir leid, aber ich kann dich jetzt echt nicht gebrauchen. Ja, sorry, ich würde gleich, aber jetzt geht's wirklich. So I'm still here, don't worry.
Hi. How is the situation in Lyon, Ralph? Everything reasonably okay?
Well, who knows? I'm at home because I was feeling shaky, and so and we seem to be beating all sorts of Italian records in terms of how many people we get infected. So.
Also in Lyon, not only in Paris.
Lyon is worse than Paris.
Oh, wow. Okay.
For once. Yeah. The worst is Marseille, and then Lyon and Paris. So who knows?
Okay.
Because Italy is still cool, no?
Sorry?
Italy is still okay.
Still okay, still okay. Schools have reopened for since the last week. Everything is okay.
No.
No, don't know why. I mean,
I'm a bit surprised by Spain. I mean, they should have had a scare.
yeah, it's very heterogeneous across Europe. Very, very strange. Okay, I see most people have come back, so I think we can start on time in 1 minute. And I'm asking good confirmation for the recording, which has just started.
Yes,
Good.
Is that what you want?
Okay, so we are all set. I think we can start. So welcome back for the second and last session of this afternoon. It is a pleasure to introduce Ralph Everest, a longtime friend and colleague who's based in Lyon, and he will give a talk about understanding the large-scale structure of interface nuclei in Drosophila for different stages of embryonic development. Ralph, the floor is yours.
Thank you, Christian. Thank you for inviting me. And I guess I have to apologize for that horrible title. So I will try to make up for that by explaining it word for word. Drosophila, well, you know, right, the little fruit fly, one of the model organisms of biology. This is where lots of research is done. Then it said something about different stages of the embryonic development. In the life of a fruit fly starts as a fertilized egg, which is a single cell, right? And then the cell starts to divide, to differentiate, and there are different stages and cycles in this process. It depends on whether you look at how this thing looks in the microscope or whether you count cell divisions, and this is an extremely well-regulated process. And so whatever we want to understand it. So then at different stages in this process, and we will see that again the second half of the talk. Then it says something about interface. So that refers to the different stages of the cell cycle, right? Sort of a cell grows and then it duplicates its genome, it divides, and then everything starts all over again. And interface is the stage of the cell cycle in between divisions. So this is, you could say, the most boring part, but it's also the part that in terms of time dominates. I mean, this is what the cell does most of the time. And so we will look at interface. Then we look at nuclei. So I mean, of course, the cell nucleus, the nucleus of eukaryotic cells, I mean, higher organisms. So it's one of the organelles in the cell, and it's sort of it's membrane bound. It contains all the genome, the chromosomes of cells. Then there is a large-scale structure of interface nuclei. So what one knows about this structure is, I mean, the two dominant effects are, the first is the existence of territories. And so that's a non-trivial thing, and it's an experimental observation that you see that different chromosomes, what they do during interfaces, they decondense, and a priori you can't see them anymore, you can't distinguish them anymore. But if you find ingenious ways of painting them, then you can really see that these chromosomes, they do not really mix, which is not expected if you use the simplest models of thinking about these problems. And the second one is an organization into compartments. This is, you could say it's orthogonal to what I just said about chromosomes. So it says something about U and heterochromatin, which I will explain in one sentence on the next slide. So there's different parts of the chromosomes, but each chromosome has both types of chromatin. And now the euchromatin and the heterochromatin, they would try to aggregate and phase separate from each other. So if you look at this kind of an organization, you would find one of these two types typically at around the membrane and the other one in the center of the cell. Okay, now comes the last word on understanding, right? So what do we mean by understanding? What I mean by understanding is that I want to use polymer physics, tools from polymer physics to understand the organization, because if you take a closer look into the system, then what you see is, to quite some extent, what we're dealing with, very appropriate for the school in Italy, is a bowl of spaghetti. Right? You have very long polymers in there, DNA, chromatin, and that has profound effects on the way things are organized. So now, instead of walking you sort of through this hierarchy of organization that starts at the double helix, and then there are histones and chromatin fiber, and so on and so on and so on, maybe what I should do is I should just sort of put down a few length scales so that we know what we're talking about. I mean, the smallest scale is something like the diameter of the DNA double helix. So here you see the picture, Watson and Crick with their famous model. And so that is of the order of nanometers. If you say, well, the other picture I just showed you, right? I mean, what is the diameter of the nucleus, depending on what kind of cell we're talking about, but it's sort of in the range of microns. Now, if you ask a different question, if you take the DNA of one chromosome and then you just stretch it out and you just ask what is the length of this double helix end to end, then this is in fact in the centimeter range. So I mean, these things are gigantic, right? These are macromolecules and they have a length which is macroscopic. If you take all the DNA in one nucleus, so now for humans, right, we have of the order of 100 chromosomes. then you end up somewhere in the meter range. And now if you do something which is totally silly, you just say, well, let me take all the cells in our body and let me just multiply this meter by the number of cells in our body. That gives us the total contour length of DNA in our body. What you end up with is something which is astronomical. That as a motivation why we should deal with long macromolecules and their properties. So now the lessons from polymer physics that sort of I will try to illustrate are the following. I mean, there's one thing which is very important for the dynamics, it's topological constraints. So that sounds like a fancy word, but it just means if you look at that bowl of spaghetti, then a priori, these spaghetti cannot do anything, these microscopic spaghetti cannot do anything that macroscopic spaghettis cannot do. And macroscopic spaghettis, I mean, they cannot cross through each other. And so then this is called topological constraints because it's the thing which is at the heart of, for example, knot theory. Then a second lesson from polymer physics is that things can become very slow because these molecules are very long. And the third thing is that even if you have very weak interactions between different kinds of spaghetti, if you like, they can nevertheless cause the emergence of a large scale spatial structure. So that's polymer physics. And then polymer physicists, they would say one more thing, and that's of course what physicists love, and I mean, I'm one of them, so I love it very much, is that if we want to understand these things, then to a very large extent, they're universal, right? They are the same for, okay, I keep saying spaghetti, but I mean, for different kinds of macromolecules, which are nevertheless microscopic, of course. So we keep saying, well, we don't really need to worry about all the microscopic details in the system. And then if we talk to a biologist, the biologist will say, oh my God, details are everything, right? And so there is a tension between these two views that somehow we need to resolve. And since this is a school about simulations, I guess I'm trying to make the point that simulations are a very good tool for doing that. So what I want to do is I'll talk first a little bit about the physics aspects. And then the second part is actually trying to look at Drosophila in different stages of development. So the first physics question is, I mean, this importance of topological constraints that I postulated for the structure and the dynamics of interface chromosomes. Now, essentially, it comes down to the question, I mean, can polymer physics tell us anything about how we brought up to a macroscopic scale? I mean, how to arrange 46 robes of length 100 meters in a box of 1 meter cube, right? So then I'm thinking about the human nucleus. Well, if I want to model this as a polymer problem, and of course, what I do immediately is I throw away, as I announced, all the details, but I just need a description of a chain molecule. And essentially, you just need to specify two things. You need to say how long such a chain molecule is, the contour length, and you need to specify how flexible it is. So typically one uses the persistence length or the Kuhn length, which says sort of under the influence of thermal fluctuations when such a molecule starts to bend. And so when we model chromatin, at least in the first part, for different organisms, I mean, we don't care so much about the details, so then the difference is just the length of these chains. So that's the structure of these spaghettis, if you like. One important point is, what is the dynamics? How can we relate results that we have to real experiments? And so what you see in that plot here is the mean square displacement of a genomic site. The blue points are data points from some experiments where you fluorescence label it, you follow its motion. And then the red points, they come out of this kind of a simulation. And by just matching the time scales, you get an idea about where you are in this kind of description. And the point to make is that this kind of a description, it's a very coarse-grained description, right? You already had talks about RNA and things like that. Well, these are atomistic models, right? And then they are simulated, and then they are simulated at the femtosecond scale. Now, these are coarse-grained models, so the units are much bigger, but it also means that the characteristic timescale is much bigger. And so that is a reason why we can simulate timescales, which are also much bigger than something that you would do for a microscopic model. Now, in technical terms, how do we do that? In fact, the model that we typically use is a very simple model. I need to model this chain molecule, and I model it as a bead spring polymer model. So these chains are composed of beads. They typically have Lennard-Jones interactions or purely repulsive versions of that. Then these beads are connected by springs. So that gives you, if you like, a linear chain molecule, but you could do any kind of connectivity if you like. Then we can tune the stiffness of these things. As I just told you, that is sort of one important aspect when one wants to map to an experimental system. And then again, this business about topology. So there is sort of the energy barrier for two chains to cross for this kind of a model is of the order of 70 kT. So that doesn't really happen in a simulation unless you force the system. So what we use is a variant of the Kramer-Grest model, and Kramer and Grest, they are famous for this kind of simulation, this kind of a picture. Again, related to these topological constraints, where the theorists were saying, okay, the consequence is that a chain actually, because it is surrounded by all sorts of other chains which block its sideward motion, it can only really move along its coarse-grained contour. And it was sort of this kind of a model and this kind of a simulation where these things were seen really for the first time. And that was a big step forward for polymer physics. So we use these kinds of things together with Angelo, and maybe you've already seen this animation here. So what we did is we wanted to simulate the decondensation of a chromosome. So we imagine we start at this point in time when the chain is dividing, when the chromosomes are distinct and condensed. And then in the next step, they can decondense. And we just said, well, okay, we're going to use these very long chains and we're going to let them decondense in a finite volume. So you just see one chain, you see a box, but there are other chains and there are periodic boundary conditions, so that sort of fixes the density of this thing. And then this initial configuration, it's very strongly folded, and then whoops, we let this thing run. If I click on the right thing, you should see this. And we let it run, and we let it run, and we let it run, right, for a very long time. That corresponds after this time mapping to something like 3 days in real time. And when you do that, and when you look at what you get in the end of this process, sort of here you see a snapshot from the last configuration. What you see is, in fact, the emergence of territories like what is seen biologically. And we can even be a bit more quantitative by saying, well, let us measure something like the typical spatial distance between two sites as a function of their genomic distance. And then what you see is the lines that come out of the simulations and the data points that come out of experiment. And the funny thing is, there are no adjustable parameters of this comparison. Right? Doesn't mean it's correct, but I mean, it's not bad. And so what does that mean? What can we do with this kind of thing? Well, it's encouraging if you want to do modeling of biological systems, right? Because it means we can make a comparison between what we get for a relatively simple polymer model and real experimental data for a biological system. Now, the second thing we can do is, well, because I mean, we're physicists and not biologists, we can say, well, do we understand what we saw, right? Can we build a theory? Can we do these things? Which means, again, this time now we're going to compare the simulation to theories or to simulations for even simpler polymer models. And maybe these two slides, they are even the most important slides in the talk, right? I mean, this is a school, and So I'm not so much talking about how you do things, but maybe it gives you an idea of why you want to do this, because it puts you into a unique position somewhere in the very center of these problems, because you can really sort of quantitatively address whether models are correct and whether theories are correct. So what one learns in terms of theory is that, in fact, you don't really have to worry so much about the motion of the chain ends, because what these polymers show is, these chromosomes show, is territorial behavior, which is something that polymer physicists know from ring polymers, non-concatenated ring polymers. They show the same kind of a behavior. It's not obvious why that should be so, because chromosomes are linear, and linear chains, usually they interpenetrate, they mix, they entangle. There's nothing that stops them from doing that. And it's only these ring polymers that don't do that. And so it's a bit strange, you can say, but why do the chromosomes remain untangled? If I say remain, well, I mean, we could say they are not entangled with each other at the beginning, right? If sort of during cell division, right, they're separate from each other, then we let them decondense, and we made the assumption that they're not internally knotted. Then what you can do is, if you map this thing to what people know about polymer physics, you can estimate the time it takes to really equilibrate the system, to build all these random knots that should be in there. And if you do that for human chromosomes, you find it's like 500 years. That's too long. And for Drosophila, that will occupy us a little bit later. It's still 5 years. So you don't need to worry about thermal equilibrium in biological systems at that anyways. Okay, so the second part is that, well, epigenetic state dependent effective interaction. Well, that's again nasty words. So far, sort of, I treated my chromosomes or my chromatin fiber, right, the spaghetti model, and everything was the same. And that's maybe overdoing it a little bit, right? Because in fact, the properties of this chromatin fiber, they are position dependent, right? I mean, obviously there's the sequence, the DNA sequence. Then there are things which come under the heading of epigenetics. So I mean, the DNA can be methylated or not. And then very importantly, it can be associated with all sorts of proteins. So proteins make up roughly 50% of the chromatin fiber. And this is highly regulated. And so what people found is that this is not random or anything. In fact, what you find is, you find a small number of characteristic types of chromatin fiber. And maybe the only one that you should keep in mind is, but that's the red one, because I'm going to use these same colors later on. Red one means these are active parts of the chromatin fiber. This is where we find actively transcribed genes. So we're not really dealing with a homopolymer. And that is not really so shocking for polymer physicists, because polymer physicists have been looking at what is called block copolymers for a long time. Polymers composed of different constituents. And they are very interesting and very exciting, because they can actually fold into larger scale structures. And the same here. So one should maybe use a block copolymer model where sort of beads of a certain color mean they represent parts of the chromatin fiber of a certain type, of the kind that I just described to you. Okay, so now we arrive at Drosophila. So I already showed you this little sketch of these different cycles of development, and I will not dive into that too strongly, because really this is not my specialty. But what we could say is, at least let's characterize what the genome of Drosophila is. And Drosophila has 4 different chromosomes, and so not 23 like us. You can feel very superior, but I don't think that makes much of a difference. And the 4th one is extremely small, but doesn't matter for us. But I mean, okay, so what we could do now is we could say, we want to adapt the kind of model, we want to develop the kind of modeling that I've shown you so far for this specific case. And we don't want to do that sort of just as a theoretical exercise. So this was part of a collaboration with an experimental group. And so what Yuki Ogiyama and Giacomo Cavelli did was they made high C experiments. So what that technique allows you to do is it allows you to measure contact probabilities genome-wide. So you get a contact probability between, I mean, any site and any site, you get data on that. And the resulting plots are of the kind that you see down there. And I will discuss them a little more, right? There's a color code. So red means there's lots of contacts, and there are lots of contacts on the diagonal, meaning there are lots of contacts between genomic sites which are close to each other along the chain. And complementary to that, we did simulations, which are essentially the kind of simulations that I showed you that I did with Angelo many years ago. The difference is that now they might have this additional coloring, which you see in the upper left corner, which corresponds to these different epigenetic states. And we know from independent experiments where to find which state. Okay, so now what do the experimentalists find when they look at their data, right? So this is data now from very early stages of development. Sort of these maps, they are symmetric. So what I will always do is I will show you just one half of this. This is experimental data, and then the simulations are going to be in the other half of this matrix. And as I told you, this technique allows you to measure contact probabilities genome-wide. And so what you see here, I mean, these little cases here, they refer to the different chromosomes. So this is the left and the right arm of chromosome 2, the left and the right arm of chromosome 3, the very small chromosome 4, and the X chromosome. Okay, so what does one see? Now, the first thing is, as I already said, most contacts are located along the diagonal, right? This is not surprising at all, but it just means that genomic sites are close to each other in space because they're close to each other along a one-dimensional chain. Right? I mean, how could they not be, right? So it's not surprising at all. Now, the second feature is what you find here. So this is now a correlation between the left and the right arm of chromosome 2. And you see this pattern. If the rest was there, would be an X-like pattern. So how can you understand that? Well, in fact, if you take drawings from like 100 years ago of what people saw in the light microscope, because for Drosophila you can see territories, then these contacts here, they just correspond to contacts between sides on these two arms, which have an equal distance from this middle thing here, which is the centromere, which is where during cell division, the machinery docks and pulls the chromosomes to the two daughter sites. And so you see this sort of V-like or U-like folding, and that's these characteristic Rabel territories for Drosophila. So now I don't know if you see this so well with the resolution, but again, here you can see an X. So now this is a correlation between the two arms of chromosome 2 and the two arms of chromosome 3 now. And just what it means is, again, in this drawing, it means these are contacts between arms on different chromosomes. And this correlation shows you that that is not random. They're not randomly oriented in this cell. Okay, so now comes the simulation. Let's see if this thing can run. So we stick in the right number of chains, the right length, whatever. The colors in this thing now refer to chromosome identity. And then we just let this run exactly as before. And we don't do this just once. We do this many times. And then we average over the results. And then what you get is, you get, again, a contact map, something that we can measure in the same way as the experimentalists, and then we can compare these two. And what you see is, this looks fairly reasonable. I mean, it's not perfect, but it looks fairly reasonable. And now we can advance, we can look at later stages in this embryonic development, And what happens there is just the duration of this interface is a bit longer. So we let the simulations run a bit more. And so there's a bit more of intermingling than before. And this still looks reasonably reasonable. And even more. And suddenly it looks totally different. Suddenly it's a bit cheating because this is data now for late embryos. So they are not on this scale anymore, so that they are almost ready to be born or whatever insects do. So these things look pretty different, right? So I go back one. So you had these rubble territories, these X shapes, very, very characteristic. Well, here you don't see much. I mean, you hardly see any characteristic context between different chromosomes. Most contacts are within arms of chromosomes. And if we want to reproduce that, and what you can see is we do it with this kind of simulation, what we need to do is we need to assume that these chains are in a different initial state, which is much more isotropic before they start to decondense.
So
Okay,
Ralph, let me remind you, you have a few minutes left.
Yes, perfect. I'm good. So now what the experimentalists do is they don't care so much about this large scale view. What they do is they zoom into these maps. And the stunning thing is really the resolution that they have with this kind of a technique. So they can zoom in and zoom in and zoom in. It's quite remarkable. Okay, so now we do the same thing. We zoom in, right? And we do this first now in the early stages of development. So you zoom, now this is one arm, I've forgotten which one from this overall map, and then we zoom even in further and essentially don't see much happening. So again, later cycle 9 to 13, not much happening. And then suddenly something happens. You zoom in this cycle 14, and what you see is you see structure. Structure means that it depends on the identity of our genomic sites, whether they have more or less than average contacts, right? So you find these, we call these TADs, where you have local zones where there are many more interactions than before. And the interesting thing is that this happens at a point in development, which is called the maternal to zygotic transition, which is in fact when these developing embryos start to express their own genes. And something happens that generates structure in the genome. And in fact, if you look at, this is now data for the late embryo, I mean, there's not even a big difference compared to what I showed you for the cycle 14. So that's something that the biologists find extremely interesting. And then I'm always nasty. So I'm saying, okay, on the left-hand side, you see the observed data. If you just average along the diagonals, that's what they call expected. The dynamic range is something like 4,000 in contact probability. And if you divide observed by expected, you see patterns, but the dynamic range of these patterns is much, much smaller. It's 16. So this average thing is what we produce. This is what I've shown you before, right? So now we have to do a bit more work to also reproduce these patterns. And as I already told you, what we're going to do is we're going to use a block copolymer model. And so now the coloring, if you like, is in terms of epigenetic identity. And you could say the green bits are heterochromatin and the red bits are actively transcribed genes. Okay, does it matter? So we put this in. So here I show you, in fact, these are very simple simulations, they just distinguish active and non-active domains. And you can't even tell the difference to what I showed you before. On larger scales, this has hardly any effect whatsoever. But we can zoom in. So we can zoom into this arm. So, okay, again, it's always the comparison, the prediction from the simulation and experimental data. and we can zoom in, and that doesn't look so bad. And we can zoom in a bit more, and that looks fairly reasonable. And this is not just an accident. We can zoom in at another position, and it still looks fairly reasonable, even though it is not perfect, because this was a model that only had a single adjustable parameter. So what can we learn from this? I mean, I would say, first of all, we can learn from this that this is an interesting polymer physics problem. Secondly, we can learn from this that I think simulations are a very powerful tool to connect physics and biology, to connect experiments, data from different systems. Now, for biology, what you can see is you get a quantitative relation between simple polymer models and massive experimental data. And from the physics point of view, well, there is sort of this relation between crumpled polymers and randomly branching polymers that Angelo was maybe talking about. So all the simulations in the first part were Angelo's, and in the second part, they were done by Pascal. And over the years, I've enjoyed collaborations with many very interesting colleagues. And with that, I thank you for your attention, and I'm happy to answer your questions.
Thank you. Thank you very much, Ralph. So please join me in thanking the speaker for the very nice talk. Thank you for being on time and also for having given such a didactical walkthrough, both phenomenology and theory, physical modeling of chromosomes and genomes based on polymer theory. So the session is open for questions. So Ralph, maybe just to get started while people write their talks, would you like to comment a little bit on the progress there has been both, let's say, experimentally and also maybe theoretically in detecting more reliably inter-chromosome interactions, which were very challenging and very noisy at the beginning, but now we're starting to get more reliable data, no, from IC maps.
Mm-hmm. Okay, maybe I should explain the problem first. Maybe if I find one of these things where I had this technique. Okay, so I mean, don't look at the details of this technique, but the way it works is you have, I mean, the cell nucleus, you throw in something that essentially glues together DNA, which is close in space. Then you cut up these bits and pieces, you ligate them, and then all of this relies on sequencing. And so if you sequence the end result of such a contact, then what you find, and if you analyze the sequence, and then if you know the sequence of the entire genome, then what you see is, oh, I have a bit of DNA which does not fit anywhere along the chains, because I've glued together two different bits which come from different parts of the genome. And that allows then to identify where these contacts are, and that's at the origin of these maps here. But then, as you know, in our cells, we always have two copies of any gene, right? One copy that we have inherited from our mother, and the second copy that we have inherited from our father. Now, of course, our mother and our father are not totally identical, but the difference is in terms of gene sequence, they are fairly small. So if you do this experiment, typically you cannot distinguish whether you have, for example, in these maps that I show you here, let's take the whole one. If this contact between the left and the right arm of chromosome 2, whether this is a contact within the same chromosome, so whether this is an intrachromosomal contact, or whether this is a contact between the left arm of chromosome 2, maternal copy, and the right arm of chromosome 2 paternal copy. And of course, if you want to understand the structure of the genome, that's an important distinction to make. So for the simulators, it's easy, right? I mean, we know who is who, so we can distinguish these contacts, but for the experimentalists, it means that they have to work much harder on the sequencing in order to identify who's who, and that is something that is beginning to emerge. And that gives, of course, additional information about how this works. Right? For example, a stupid question, are the two copies, are they close to each other in the cell or not? And that's something that one could learn from this.
Thanks a lot, Ralph. So there is a question from Kathari Azizi. So can you comment about the temperature, how temperature is considered in the model, and how much is it? So in terms of model temperature and probably real temperature, I believe.
So when we build the model, let me see if I find this thing. I mean, I told you that there is this length scale here, which says when a chain can start to bend under the influence of thermal fluctuations. And so the mapping is such that you just get that length scale right. Then you don't have to worry about temperature. In the same way, temperature would tell you how fast you can diffuse. But since we use this mapping to real time, we don't have to worry about temperature as such. Now you could ask an additional question, which is about biology, right? Now, for us, temperature is always fairly constant, right? I mean, we have a very narrow range. Out of that range, we're simply dead. For other organisms, that is not true. And you could ask, does it matter whether or not you were at 270, whatever Kelvin, or at 290, whatever Kelvin? Which I always think for biochemistry is it's an interesting question. I think for these problems, not so much.
Okay, thank you.
It's not a gigantic effect.
We have a second question from Isabel Louise Grothaus, who's asking to comment about, I think, the spirit of the simulation. She's asking whether it's more in the MD spirit or I believe the Monte Carlo spirit. She's asking, how do these polymer simulations actually work or are computed? Similar to MD, or are they like coarse-grained? I think she wants to learn more about the stochastic dynamics.
Okay, so now technically, this is molecular dynamics, but it is not molecular dynamics with an atomistic force field. So if you interpret what we use in terms of a force field, you could say we use an interaction potential for point particles, which is very popular for argon. And then we connect these beads with springs, you could say essentially harmonic springs. So it does not really have much of a microscopic interpretation on the scale of this model. And the justification for doing such things is this point that I brought up somewhere, but I don't remember where it is. I think a little back. This business of universality. So what polymer physicists know is you can do experiments with different kinds of polymers. And if you look at very different kinds of polymers at different temperatures, if you look at them using the right units, then in fact they all behave in the same universal way. That is true for properties which depend on the large scale structure of these systems. And that gives you a lot of flexibility in how you want to model them. right? If universality means that things that are characteristic of polymeric systems do not depend on the microscopic details, it means in terms of formulating models for polymers, you can take anything which is computationally convenient, or it's the same for doing calculations. If one wants to do calculations with polymers, I mean, the simplest theories are random walks on a lattice, and then if you solve that model, you end up with these Gaussian distribution function that Professor Icahn was talking about. Only they mean something totally different. They mean something about what is the typical conformation of such a molecule, but it turns out the mathematics is the same as the mathematics in diffusion. And so from a technical point of view, what we do is MD simulations, but these are coarse-grained models. They're not trying to capture what is going on sort of on the scale here of individual atoms. Because then we need, I don't know how many orders of magnitude more powerful computers to do the same thing. And in the end, they will probably behave in exactly the same way. But there's a bit of understanding in making that decision, what matters and what does not matter. So this is a top-down approach in terms of modeling. Right? You start with identifying the coarsest features, the essential features of the model, and then you add detail to it. Instead of saying, well, I try to represent my system on the level of atoms, then I run MD and I see what I get. Right?
Very well. Okay, so Isabel Luis thanks you for the answer. Other questions? So Raf, maybe would you like to comment a little bit on what you think are the challenges, either the next ones that should be tackled or those that would be like dreamlike to be able to tackle in several years?
Yeah, let me add one thing, which is to say the slide I have after the conclusions. Which is here. I mean, there's more than what I've told you. Sort of the cell nucleus is an active system. And there are many things going on that we have simply ignored. And now, there are two ways of thinking about it. One way is to say, that means everything we've done is wrong. And the other way is to say, it's interesting that something which is so much more complicated comes out to behave in the same way as a much simpler system. Okay? So the interesting or very interesting mechanism, which is characteristic of these polymers, so these chromatin fibers in the nucleus, is what has now been established as being loop extrusion. So there are, and it's related to sort of this next stage in the story, right? I went to just decondensing, but at some point you have to replicate, and then you have to condense. And one thing you have to do on the way is you replicate these long polymer chains. After you've replicated them, they are totally knotted with each other. And you have to separate them from each other. And one good mechanism for doing that is loop extrusion. That is, you sit somewhere and then you pull out the length in between. And there's a lot of this activity going on in these systems. And then the question is always, A, can one model it? B, I mean, there's always the question, if somebody shows you results which are so much more impressive than the ones that I've shown, right? But then if they've used 1,000 parameters, then I'm less impressed, right? So there is something in, yeah, it's sort of a bit of Occam's razor approach. But the physics of these active systems is very interesting. And the other aspect that I mentioned, right? I mentioned these effective interactions between different kinds of the chromatin fiber. But on the scales where we model them, they are relatively weak. But if one goes to smaller scales, then they can become much stronger. And in the way biology functions, that also has effects. So there's much more to be understood. I would still say it's reasonable to have this top-down approach to start from the coarsest feature and then to see how much more you can add and get back what you had before and get something in addition which you didn't have before. Sort of in my talk, I did one step, I put in a bit of identity along the fiber, but there's much more along these lines that needs to be done. Right, and all of this is implicated in gene regulation, which is the big question. Right? And all of our cells, they contain the same genome, but our cells are doing very different things. And how does that work? That's a fairly non-trivial question. Structure contributes to gene regulation.
Thank you, Ralph, also for giving us a peek into the challenges and the physicist perspectives, how to tackle them in a convincing way, to use models to identify the prominent underpinning physical mechanisms, no? Without getting lost in details and overfitting. So I think we are at the end of the session. So I really wish to thank Professor Everest and also Professor Eichhorn that we heard before. I'm really grateful for them, no? And as we all are, for having taken time out of their busy schedule to give us really a flavor for fundamental topics that should be in the basic knowledge of everyone, very fundamental also for these second school on numerical methods applied to physical systems. So thank you very much again to both Ralphs. Thank you.
Thank you. All right.
All right.
My pleasure.
Okay, so I think we can stop the recording now. Great. So really, many thanks to both of you. It was an excellent afternoon, very, very stimulating. I think you both hit the audience at the very right level.
Okay, very good.
I'm happy to hear. Great, thank you.
Yeah, because I was scratching my head what I should do actually.
Yeah, I mean, giving these talks is always talking in front of a mirror, no, in some way, but you're getting lots of feedback. You can see in the chat box, people thanking. It's, yeah,